Okay. So let's start the class. Last class of discuss korisilo, we were talking about what was the main topic that we have discussed? Development of the microspores, I think. Na? We are discussing about something called as microsporogenesis, and what we remember, microsporogenesis mane ki? The formation of the microspores is called as microsporogenesis. Who forms the microspores? Who forms the microspores? It is a microspore mother cell, right? Microspore mother cells are also called as pollen mother cells. They are also called as sporogenous mother cells, you can say, right? I think we remember about that anther, dithecous anther. Then we have talked about cutting a TS of the anther, and then we have discussed about something called as a archisporial cell, a parietal cell, and a sporogenous cell, right? Who forms this wall of the pollen sac? It is a parietal cell, and who forms the pollen grains? Ultimately, it is the sporogenous cell. Sporogenous cell would behave as spore mother cells. They will undergo meiosis. They would be producing tetrad of microspores. As far as the arrangement of the microspores is concerned, we see different types of arrangement. We see different types of arrangement in different types of plants, right? And among all the types of arrangement, the two of them that were most commonly seen, one in case of dicots and the other in case of monocots, was isobilateral and tetrahedral condition. This is something that we have discussed, and together with that, what we have discussed is when the pollen grains are formed, they are formed in the form of a tetrad. Everything joins together, right? But prior to pollination, what happens? The tetrad breaks down, the pollen grains separate, and then only they are ready for pollination, right? So the next thing that we are going to study is what happens to the pollen grain, or we will be studying something called as the development of the microspores, right? We have last class of porizolo, formation of the microspores. Aji ki porim, development of the microspores. When we talk about development of the microspores, we can also use a word saying that germination of the microspores. This means the same. When I am talking about germination of the microspore, eta line ami likhisilu no, pollen grains or germination kenuka dhoroner hoy? Is it in situ germination that they show, or ex situ? In situ, so we understood that they will be germinating when they are inside the microsporangium itself, right? So etia microspore to keneke develop hoy, mane microspore ki hoy? Nothing but a single cell, right? Once they will start dividing, so obviously at the first time they will be forming two cells. So that is the process that we are going to study now, right? So heading korima, development of the male gametophyte. Okay, the first question, male gametophyte mane ki? Male gametophyte mane? Which structure are we talking about? So everyone sure about the answer, or is there anyone who doesn't know what a male gametophyte is? So anazanile does not matter. It's good to clear the concepts. It's very good, but provided everyone know about it. Male gametophyte mane ki? What is the other name for male gametophyte, or which structure is called as a male gametophyte? Microspores. Phyte mane leaf nohoy. Phyte mane plant buli koy, that organism. Phyll mane leaf, right? Microsporophyll paisilu no ami. Phyll is equal to leaf-like structure. Phyte, plant or organism. Plant aro. Jar bhitorot tumar gametes kita thakibo, right? So we are talking about what? Tumi jana? Ki hoy? Male gametophyte mane ki? Microspores. Microspores are eta naam? Pollen grains, right? Okay. So jot niki male gametes kita thake, tak ami ki buli ko? Male gametophyte buli ko. So we are talking about ki? Microspores, which is also is equal to ki? Pollen grains, right? A point to right. So, when we talk about the development process, the entire process is divided in two stages, that is we will be talking about pre-pollination development, what are the changes that occurs in the microspore prior to pollination and after pollination. So, what we will do is at first we are going to draw a diagram, and from the diagram itself we are going to understand the thing, right. So, next আমি diagram আঁকিম. Structure of a microspore we have already discussed, yes. It's a rough diagram that I am drawing. So, we are talking about the same spore, do not go by the size of it. Obviously, the size remains the same. এইটো লাষ্ট ডায়েগ্ৰাম। ফ্লাক্সবোৰ আৰু দিব পাৰে, কিন্তু এপিনলৈ নাহে, অকল এপিনলৈ থাকে, ৰাইট? এই গোটেইবিলাক পইণ্টছ মাইক্ৰস্প'ৰৰ All? Okay. So we are talking about what? Development of the male gametophyte. When we talk about the male gametophyte, what we are talking about is a microspore or a pollen grain. When we talk about a pollen grain, structure of a pollen grain is something that we have already studied. When we talk about structure, it has covering exine and intine, we understood, right? So exine, exine continuous ne pore ase, obviously pore ase. What is the function of a germ pore? It is that area through which the pollen tube would be formed or will come out. Right. So what happens to the pollen grain is at first they will be dividing as usual, but the point to be noticed here is when the pollen grain divides, they will be forming two cells. One is a tube cell and the other is a generative cell. Kintu diagram to equal dekhisa na unequal dekhisa? Unequal, right? Tube cell, generative cell. So what they will be doing is they will be forming two cells, kintu not two equal cells. One a smaller cell, the other a bigger cell, right? Next is tube cell, tube nucleus. We understand since we are talking about a cell, so obviously here cell to obviously nucleus thakiboi, right? Next point on Q is what you see is callose deposition in and around the key generative cell. Next point, next diagram on ki dekhisa, so because of this callose deposition in and around the generative cell, this is something that we observe. Now what is the difference between this diagram and this diagram? First diagram to buji paisu. We are talking about two unequal cells. Difference is, okay, this is one point of difference, degenerated nucleus, because this nucleus will start breaking down, right? Yar pora start hobo, kintu we are showing it in this diagram. But the point is, A2 diagram or difference ki, yate ki ase, je duta sharp cells ase, divided into two cells, right? But in this condition, what we see, it looks as if the generative cell has been floating inside the tube cell, yes or no, right? So up till here, we are talking about pre-pollination, and this diagram talks about post-pollination. Is up till here understood, right? So when we talk about pollination, we know the process, the pollen grains will be shed off, right? Kot poribo stigma go apply. This is a condition that you will see. What happens in the stigma is, in the stigma, the pollen grains will fall and they will be absorbing the water that is already present in the stigma. Jikono bostu eta grow koribo lagile na pani lagibo, right? So stigma already water was there, pollen grain will fall because of pollination, they will be starting absorbing the water, and as a result what happens, germ pore through e bitorer cell to ki asile? Tube cell. So tube cell will come out to the germ pore and ultimately they would be forming key pollen tube. So who forms a pollen tube? It is a tube cell that forms a pollen tube, right? Once this content, the entire content comes out from here, so obviously generative cell to ahibo. Kintu prior to this, generative cell e ki kore, it divides and forms two cells and they would be acting as ki two male gametes. Now A2 diagram par question ki ahe, the position of the generative, the position of the tube nucleus. Tube nucleus to agote yate asile jitia ahi jai no, then the position is always at the tip. Is the point clear? Okay. Next, something to discuss from here is, when we are talking about this pollen tube, we will be studying a line saying that the growth of a pollen tube is apical. What do you understand by that line? The growth of the pollen tube is apical. Tarmane, it grows by its apex. Right? So if this pollen tube grows by its apex, so maximum cytoplasm ko thakibo, but living cytoplasm khini ko thakibo yate, right? So what happens is, as the pollen tube keeps on growing, now we know that at the first time itself, pollen tube dal to iman dikhol nasile no, it was very small and gradually it started growing, right? So jetia horu asile, tip to yate asile, gradually jetia dangor hoi gol, cytoplasm khini kole move korile towards the tip. So this old cytoplasm and the new cytoplasm got separated by callose plugs. So ultimately callose deposition hoy jabo, callose deposition hoy jua lage jeta deposition bor barhi goi thakibo, ekhini bostu pura area to dead hoy jabo na jay. So living area kunkhini, so thick cytoplasm kot thakibo ba living cytoplasm ba new cytoplasm kot thakibo apex hoy. Is the point clear? So we will be writing the points about it. Diagram hol no. কিছুমান পইণ্টছ লিখিম below the diagram লিখিবা From the diagram itself, we can understand these points, right? Okay, this is, so division kenekwa, unequal, right? Next is tube cell is also called as, it is called as a vegetative cell, and generative cell, since it forms the male gamete and tube cell does not form the male gamete, so it is called as a vegetative cell. From reproduction point of view, gametes jehetu generative cell form kore. So reproductive cell, actually who acts as the reproductive cell is the generative cell that acts. right. Baki rest of the lines I think it's understood. Okay. So, next we come to two words, bicell stage or two cell stage and a three cell stage. We are talking about ki pollination, right? Shedding of pollen grains is nothing but pollination. Pollen grains will have form korile, develop korile, but germinate korile, and what we have seen during the process of development that the pollen grain has divided and formed in two cells, that is a tube cell and vegetative cells. Once the two cells are formed, they are ready for pollination, and that is a condition that is commonly seen in most of the angiosperms. So, this is a point that is important, right? Maximum angiosperms or pollination kuntu stage hoy two cell condition, bicell condition, and by the word two cell, what are we talking about is formation of this and this. But sometimes what we see is 3 cell hobo, tar pisothe pollination hobo. And when we talk about 3 cell, obviously tube cell to thakibo, the generative cell will divide and form the 2 male gametes. Ultimately, microsporode keta cell ase tinta. So, after that structure only the pollination will take place. However, which one is common? 2 cell condition, right. So, question has been asked about the common condition in angiosperms. So, ekini whole pollination বা pre-pollination or okay. Next likha post-pollination development. Is this over? Post-pollination, as far as the description is concerned, we have already spoken about it. What did we see? The pollen grains will get shed and they will fall to the stigma. In the stigma, what they will do? They will be absorbing the water. They will absorb, once they absorb the water, they will swell up, and the entire contents of the tube cell through the germ pore would come out and ultimately would be forming the pollen tube. As far as the pollen tube is concerned, what is the position of the tube nucleus? Tip of the tube. See, that's all. As you are writing the lines, see whether you have understood or not. We have already explained, right? So we are just writing the notes now. So from all these points, again, this is something that has been explained. This one, the growth of the pollen tube is apical. We understood this word. It grows by its apex. We have another word that is called as chemotropic. What is chemotropic? Movement because of the attraction of certain chemicals is called as chemotropic, right? Kiba chemical secretion of karne jedi hi grow kore, right? Tak ami ki boli ko chemotropic, right? Amar kisman words ase jeneke thigmotropic, chemotropic, kisman light sensitive plant ase phototropic hoy, right? So what happens is, actually what we are talking about is, we are talking about this as the stigma-stylon ovary, right? Yate ki hoy pollen tube, pollen grain to form kore in a bicell condition. Pollination hoy jay, pollination hoar pisot pollen tube to form kore, right? Pollen tube to ki hobo a stylor through penetrate kori pelai ovule to pabo lagibo. So as it is growing, it has to grow to reach here, right? So growth ki ho karne hoy by certain types of chemicals that are being secreted, and because of that chemotropic. Later we will study that when we are talking about this pollen tube, no, pollen tube what's the important structure ki? Male gamete gita. Male gamete gita, it has to reach the egg that is present inside the ovule, right? Now we will be studying that pollen tube, no doubt it has to come and fuse with the, pollen tube does not fuse, the male gamete will fuse with the egg. In a pollen tube, egg to port ase janibo to lagibo, right? So we will be talking about certain structures that will help in the process. So by secreting certain chemicals, since chemicals play a role, we are using the word ki chemotropic, right? Ekin lage kiba problem ase niki? Okay. Next, pollen tube dalor okoman information janibo lagibo. So likha zones of a pollen tube. Ba pollen tube buli heading kora. It's better to write pollen tube. So when we talk about the pollen tube has been divided into certain zones. We have 3 zones, the growth zone, the nuclear zone and the vacuolization zone. Wherever the vacuoles are present and the callose plugs are present, I think the information is written, right? Growth zone kunkhini? And what is the function of the callose plug? It separates the inactive cytoplasm from the active one. And which part is considered as active cytoplasm? Jot niki male gametes kini ase aru growth or apex to. So up till here, what we have discussed is we have talked about the male reproductive part of the flower. Since we are talking about reproduction, we are concerned with the male part and the female part, right? We remember those words called as megasporophyll, megaspores, right? Megasporangia, sorry, microspores, microsporophyle and microsporangia. Whenever we are talking about the male part. Now, since the pollen grains are ready, the male gametes are ready, what next? Tahate go play ki koribo laibo, fertilization koribo laibo, prior to that pollination hobo laibo. But since we have completed discussing the male part, next the female part, right? So heading koriba, prothome dekhi likhi do ase, oh likha hol no, likha megasporophyll, heading koriba megasporophyll. Megasporophyll mane, we are talking about which part of the flower? Megasporophyll. What is the other name? Gynoecium, yes. Gynoecium is the entire lot that is called as gynoecium, right? The individual member is called as carpel or pistil. So what is the megasporophyll? Nothing but the carpel or the pistil. Okay. So whenever we talk about the carpel or the pistil, we know the parts of a carpel. Kimanta part thake, tinta part thake, sigma, xylem, ovary, right? So we'll be writing that part. Megasporophyll, megasporangium likha. Duita bostu ekelege cover hoye jabo. Right. So we have to talk about this part, and especially we are concerned with the egg code thakibo, ovular bhitor, ovule code thakibo, ovary bhitor, right. So write this point before discussing the ovule, the egg, some points about the female reproductive part that we have already studied in morphology. Okman ami pohilo, refresh. Ekini ho gol. So before starting this point, অকমান morphology আমি কিছুমান বস্তু পঢ়িছিলো এইখিনি মনত আছে নাই চোৱা So whenever we talk about the female reproductive part, we are talking about the gynosium, right? In a flower, okay, gynosium individual member ki pistil, in a flower pistil kiman thakibo? What would be the number of pistil in a flower? No, it depends on flower to flower, right? In certain flowers we have a single pistil, in certain flowers we have multiple pistils. When we talk about a single pistil, we had a term that we have studied in morphology. Single pistil thakile, that flower would be called as ki? Monocarpillary বা monocarpillary, right? আৰু হেতু কি বুলি কম poly বা multicarpillary. Next, যদি বহুত বিলাক carpels আছে, all the carpels would remain free from each other or they will remain fused. Remember the two words? Apocarpus and syncarpus, very good. Apo kundu or syn kundu? Syn to fuse or apo to free. Ha. লিখি লোৱা এইখিনি হল megasporangium পিছে এইটো word integumented, integumented, the presence of integuments, right? What is that? I'll explain. Just write it down first. Nikhilwa will be discussing the points. Over? Okay. So coming to the first point, in the ovary, so when we talk about the female reproductive part, the ovary is an important part. Ovary bhitorot ki thake, you have an ovarian cavity with one or more chambers or locules. Do you remember this word when you have studied morphology? Remember something called as placentation, right? So placentation kenuka asile, marginal placentation asile, basal placentation asile, etc., right? So placentation mane ki arrangement of ovules in a ovary is called as placentation, right? So now the question is, ovules kot thake, ovary bhitor thake, right? Kintu ovules ovary kuntu part or thake, bhitor thake, it's understood, right? So whenever we are talking about the ovary, no, inside this ovary, this part would be called as locule, or it is called as chamber, right? Looking at this diagram, this is the ovary. Keita chamber ase? Obviously one chamber. Kintu aru eta jodi ami eneke eta ovary ako, and we are talking about this like this, then tar chambers kiman thakibo? Tinta chamber thakibo. So ovary bhitorot ki thakibo? Ovarian cavity thakibo, and that is what is called as locules. As far as the number of locules or chambers in an ovary is concerned, it depends on plants to plants, right? So we have words, or you all have studied words like unilocular ovary, right? Bilocular, trilocular, etc., right? So depending on a septa kon thakibo na thake. So if this is a ovary, tar bhitorot ki thakibo libo? Ovules thakibo libo. Ovules kot thakibo depends on the position of the placenta, right? So again, placenta ki hoy? It is nothing but a group of parenchymatous tissues, or in a non-proper definition, you can say that's a seat of the ovule. Jote ovules bilak thake, hekini jeka ami actually ki boli ko? Placenta boli ko. And placenta is a group of parenchymatous tissues, and what is the most important function of placenta is to provide nutrition to the developing ovule, right? So ovary bhitorte jote placenta thakibo, tate ovule thakibo. Suppose etu ovary je placenta iyat ase, therefore tumi ovule kini iyate paba, right? So accordingly, we had you all have studied different types of placentation. For example, do you remember this diagram? Marginal placentation in case of P, right? Etia kotha to hol, why are the ovules located in the margin of the ovary? Because the placenta is present there, right? Similarly, if we are talking about this one, exile placentation seen in case of tomatoes, right? Exile placentation or akko ovules bur yat kyo ase? Karon placenta yat ase, right? So that is something that we have studied in morphology, so we will not be discussing here. So we understand inside the ovary, what you see is chambers or locules, clear? Okay. Next, inside the ovary, we understand the ovule is present, right? Ovule or beleg naam ase. Megasporangium is equal to ovule. Ovule buliu kobo pora, megasporangium buliu kobo pora, integumented megasporangium buliu kobo pora. Kintu the proper definition or the proper word that defines a ovule is integumented ovule, right? Eta structure, he structure to ovule to tamane ki মেগাস্পৰেঞ্জিয়াম আৰু মাইক্ৰস্পৰেঞ্জিয়াম পঢ়িছা তোমালোকে আগতে পঢ়িছা মাইক্ৰস্পৰেঞ্জিয়াম মেগাস্পৰেঞ্জিয়াম মাইক্ৰস্প'ৰছ থকা ঘৰটোক কি বুলি ক'ম মাইক্ৰস্পৰেঞ্জিয়াম মেগাস্প'ৰ থকা ঘৰটোক কি বুলি ক'ম মেগাস্পৰেঞ্জিয়াম ৰাইট so we are talking about the presence of egg এগটো ক'ত থাকিব অভিউলৰ ভিতৰত থাকিব ৰাইট so অভিউলৰ ভিতৰত এগ থাকিব সেই এগ থকা ষ্ট্ৰাকচাৰটোক অভিউল বুলি ক'বা নে মেগাস্পৰেঞ্জিয়াম বুলি ক'বা We have used the word megasporangium when we have studied about the plant kingdom, right? Whenever we talk about the presence of egg ক'ত থাকিব ঘৰ এটাত থাকিব ৰাইট সেই ঘৰটোক আমি অভিউল বুলি কেতিয়া ক'ম যেতিয়া মেগাস্পৰেঞ্জিয়ামৰ কাভাৰিংছ থাকিব বা ইণ্টিগুমেণ্টছ থাকিব যদি মেগাস্পৰেঞ্জিয়ামৰ কাভাৰিংছ নাথাকে we will be using the word megasporangium and not a ovule no doubt it means the same right megasporangium is equal to ovule কিন্তু ovule word to কেতিয়া ক'বা কোনটো megasporangiumক ovule বুলি ক'বা যেতিয়া megasporangium to ka integuments থাকিব and megasporangium is something that is present in pteridophytes is present in gymnosperms etc but ovule is present in angiosperms is the point clear okay so as far as integuments are concerned আমি হেবিলাক draw কৰিম অলপ পিছত structure of the ovule this is something that's understood এটা ovule থাকিলে এটা seed হ'ব বেছি ovule থাকিলে বেছি seeds হ'ব next coming to this point we'll be talking about different types of ovules present in angiosperms around 6 to 7 types of ovules you see in nature right and out of these 6 no the most common is supposed to be anatropous so ovules bilak different types jeneke orthotropous ovule hobo pare anatropous ovule hobo pare hemitropous hobo pare campylotropous hobo pare so keneke keita dekha pam sota out of that common kuntu anatropous as far as anatropous ovule is concerned okay before going to anatropous etu word janibo lage orthotropous ovule to tumalke okkoman janibo lage karon etu bostu tumalke gymnosperms ot pohisila gymnosperms bilakor the most common type of ovule that you see in gymnosperm is orthotropous the most common type of ovule that you see in angiosperms is anatropous angiosperms ot kiman types of ovules thake 6 types কিন্তু সবতছে কমন কোনটো anatropous gymnosperms ot কি থাকে orthotropous remember this one orthotropous is also called as straight ovule তার মানে কি ovule বিলাকৰ structure akile বেছি clear hobo ovule বিলাক কেনেকুৱা হ'ব পাৰে heard about a word called as micropyle এটা ovule কি থাকে micropyle selaja থাকে না covering থাকে integuments so micropyle কি হয় akim obviously akim you know micropyle is that area where the integuments are absent it is that area through which the pollen tube will penetrate কারণ ওভিউলৰ ভিতৰত pollen tube to যাব লাগিব তেতিয়াহে তোমাৰ মেল gametes fertilize কৰিব right so যোনটো এৰিয়াৰে যায় that area is called as the micropyle now talking about the position of the micropyle you have different types of ovule যদি if you are talking about an ovule like this তেতলা এটো যদি ovule হয় এই তলৰ এটো সাইড কি বুলি কম micropyle right so if the micropyle faces downwards it is called as anatropous right কিছুমান ovule এনেকে থাকে এটো কি হ'ব micropyle যদি if it's facing upwards it is called as orthotropous so we have different types of ovules and depending on their curvature কেনেকে movement to hoise যদি ওপৰলে চাইছে নে তললে চাইছে 180 degree movement কৰিছে নে 90 degree movement কৰিছে নে 360 degree movement কৰিছে depending on that we have 6 types of ovules right and one among those 6 common key anatropous we have a slight idea about anatropous the structure looks like this right এটা নেক্সট লিখা আমি ডায়াগ্রাম আঁকিম স্ট্রাকচার অফ এন ওভিউল হেডিং করা over. So, since we are talking about the ovule, right, we will be talking about the structure. Now, it is a diagram to Akisu. Detailed study we have to talk about this, because from reproduction point of view, obviously the egg is important, right. So, when we will be studying about this structure or the ovule, apart from all the other parts, right, we will come across a word called as 7-celled 8-nucleate structure. Ovule to ki hoy 7-celled 8-nucleate, no? Bakibur sob padiason ekhini jeka jodi soa cells keta ase 1 2 3 4 5 6 and the entire 1 7, kintu nucleus keta ase 8 ta. So, ovule would be best described that 7-celled 8-nucleate structure. Ekhini ami pisat porim. As of now, whenever we are talking about ovule, first what is the type of ovule we are drawing? Since the micropyle is towards the facing towards the ground, anatropous ovule. Understood na? Okay. So, this ovule would be having the coverings that is called as outer integument and inner integument. Angiosperms of hair integuments dukhon thake. Ovule is something that is present in gymnosperms also, kintu tat ki thakibo only single integument thake. We will be coming across words called as bitegmic ovule, unitegmic ovule, etc., etc. Right? As far as the ovule is concerned, after fertilization, ovule gets converted into dash. After fertilization, the ovule gets converted into obviously seed. Jodi ovule to a seed form kore, tete integument se ki form koribo. Jeneke we have structures like ovary, ovule, etc. Prior to fertilization, it's the ovary, it's the ovule. After fertilization, the ovary gets converted to a fruit and the ovule gets converted to a seed. Okay, understood. Now, we understand that the ovule has the integuments. So, tahate integuments will get converted into what? Seed coat. Right? Okay. So, they will be forming the seed coat. We generally see two seed coats, testa and the tegmen, and properly there is sabo bisarisa kabuli boot dekhisu no. Kabuli boot to sabasa you have a outer cover that's brown in color, bhitor ekon white filamentous dekha baba. Right? So, integuments would ultimately form ki seed coat. We will be writing about that. Next, we have the central cell, the egg apparatus and the antipodal cell. Egg apparatus 3 cells, antipodal cells 3 cells, details pisot korim. Right. Next, micropyle we understood, it is the area where the integuments are absent, it is the area where generally the pollen tube will penetrate. However, we have exceptions to this statement. Sometimes pollen grains are pollen tube are seen penetrating through the integuments also. Sometimes it is seen that it penetrates through the cella gel area also. Kini hebilak hol exceptions. Commonly ki hoy micropyle is the area through which the pollen tube will enter. Right? It is that area where the integuments are absent. Next, we have a structure called as funicle. What is a funicle? The stalk of the ovule, tharidal. Lichi khaute ami tolodal thari dekhisu no. So, that stalk is called as funicle. Sometimes what happens, the funicle extends somewhat to the body of the ovule. That part would be called as raphe. Extension of the funicle, extension of the funicle over the body of the ovule is called as raphe. Next, we have a word called as hilum. What is hilum? The point of attachment of the funicle and the body is called as hilum. Right? We will be writing. And next, ovule to kot thakibo, jo placenta thakibo. Placenta kot ase, ovary ase. I think it's understood. Right? So, we will be writing the points about it. So, first likha. Diagram to over is a musiba. Oh, we did not explain nucellus. আৰু লিখা আছে নেকি বাকিকেইটা পাৰ্টটো বুজি পাইছোঁ So, coming to the word, I think it's over, right? So, nucellus point to buzibo lagibo. First, is it nucellus or it's nucleus? Obviously, it's nucellus and not nucleus. Second, what is the function of the nucellus? To provide nourishment to the developing ovule. Is the point clear? Right? Next is nucellus ki hoy within by inner to the integuments. Originally, you had a group of cells, those were called as nucellus. One of the cell of the nucellus became big and behaved as an ovule. Is that point understood? Inner to the integuments, you had a group of cells. Tamane ki prothom dinai to ovule structure enukwa nasile no moi ami jane ke when what we have studied this when we were talking about the development of the microspores, we have started a discussion from the anther. Kuntu anther loisile young anther. Young anther or prothom dinai pollen grains asile niki nasil no thik tenike if you are talking about this structure prothom dinai enukwa asile niki nasile. So initially ki asile je we were talking about a structure like this jot niki tumar integuments thakibo integuments or bhitorot jiman bilak ebilak bostu it's a collection of cells. This is something that you see at the first, right? So a gutei collection of cells of ami ki boli ko nucellus. Inner to integuments, you have a group of cells and these cells are called as nucellus that are parenchymatous in nature, right? One of the cells, bahut cell ase, one of the cell of the nucellus that lies towards the micropyle will enlarge and ultimately it will behave as actually ovule nohoy pisot ami porhim megaspore, right? As of now, who forms the ovule? Nucellus forms the ovule, right? So nucellus ki hoy collection of cells. Eta cell dangor hobo kot asile micropyle area. Eta cell jete dangor hobo etu dangor hobo goi thakibo no karon it has to form ki 7 cell. Originally ki asile single celled, right? Nucellus or when we talk about this as a nucellus, it was a single cell. Ebilak gutei bilak single cell, right? Eta single cell dangor hobo, right? A single cell to kiman cell hobo lagibo 7 cell hobo lagibo tamane he divide koribo, right? Tamane we can say this cell has to grow, right, to form 7 cells. If it has to grow, it requires food. Obviously kiba eta bostu dangor hobo lagile etak to khabo lagibo no. So what happens is since one cell will enlarge and behave as a megaspore, the other cells at that time will die off, degenerate, and they would be giving their nutrients to the developing ovule. Is the point clear? Right? So etai dangor hobo bakibur naikia hoi jabo tahantor contents bilak kune absorb kori lobo etue, and ultimately this structure will get converted into this. So what is nucellus? It is a group of cells, right? And what is the function of the nucellus? One of the cell of the nucellus would be forming the ovule. Clear? Okay. Etu diagram musu. Next likha point number 4 heading koriba integuments. So, type of ovule seen in gymnosperms unitegmic, and we understood the ovule will be having one integument, right. Angiosperms of ki thake bitegmic condition thake. We have something called as tritegmic also. I think the meaning is understood. Tinikhon integuments thakibo, kintu obviously exceptions ebilak kot thakibo. Asphodilus is an example of a monocot of angiosperm. So, angiosperms are commonly obviously bitegmic thakibo, kintu sobote exceptions thakibo, no. And sometimes you might see something called as a ategmic, where the integuments are absent, right. So, integuments absent is called as ategmic. However, from examination point of view, this too is important, right. Over? Okay. So inside the ovary, what you will see is collection of cells, that is called as nucellus, right? Certain ovary will have poorly developed nucellus, certain will be having well developed nucellus. So you need to remember these two words, question directly a word to or a yar meaning to word question hai, right? Ovules with poorly developed nucellus is called as, and with well developed nucellus is called as far as the examples are concerned, not important, only the word and the sentence. Understood? Okay. Next point about the nucellus. Normally the nucellus get completely absorbed during embryo development. What we are talking about is, all of agote jintu diagram dekhalu no, nucellus ki asile groups of cells, gutei cells bilak survive koribo ne only one survives. Only one survives. What happens to the rest? It gets absorbed. Tamane ki nai kya hoi jai. Is the point clear? So we are talking using a word called as embryo development. Ami ki pori asilo, ovule pori asilo. Ovule to ultimately it would be considered as an embryo provided it has certain structures. Integuments thakile ovule buli kobo, right? Aru kiba kibi structures thakibo lagibo, zygote thakibo lagibo, pen thakibo lagibo, tetiale ami ta ki buli kom, embryo. So depending on stages of development, the same structure sometimes would be considered as an ovule, sometimes it would be considered as an embryo. Prior to fertilization ki hoye ase ovule. After fertilization ki hobo? Seed hobo, that could be considered as embryo also, because that is the structure from where the baby plant will arise from, right? So we are talking about the same thing, right? So nucellus or ki hoy, gute nucellus bilak absorb hoy jay. So fertilization of pisot, so ovule ase, nucellus ase. Nucellus gute kini naikia hoy gol, eta cell matro ki hoy gol, ovule hoy gol, he cell to ki hoy jabo, seed hoy jabo. So jetia seed dekha pam, tetia nucellus dekha paba janu. Nucellus asile collection of cells, tare gutei khini ki hoi gol, degenerate hoi gol, thakil ki eta cell, he cell to ki hole convert hoi gol, seed hole. So jetia tumi seed dekha paba, then nucellus dekha paba niki, he group of cells kini dekha paba niki, nupua, right? So in general, but commonly observed character of angiosperms is jetia seed formation hoy, nucellus to naikia hoi jay. Kintu we have exceptions. Right? We have a certain type of a seed that is called as perispermic seed. Perispermic seeds are those seeds that will have persistent nucellus. Persistent mane ki hodai thake. Normally ki hoy nucellus nai kya hoi jay, okol ovule to he seed hoi jay, right? Kintu kisuman seeds on tamane seed formation or loke nucellus to thakiboi, gutekhini mori na jay. So junbilak seeds or nucellus to dekha pao, those seeds will be called as ki perispermic seed. So from this part, what is important is this word, seeds with persistent nucellus is called as, that's the first word. Second, obviously the examples, right? And perispermic word to lagibo. Next slide, this is something that we have explained. What we said, one of the cells of the nucellus towards the micropyle becomes big, right? We have discussed this structure. That ki hobo, eta cell towards the micropyle will become big, right? This cell will behave as ki megaspore mother cell. So obviously it will undergo ki meiosis and ultimately it will form ki megaspores, right? So megaspores ki hoy, ovule, right? So ovule kune form kore, nucellus e, nucellus or cells bilake behave as ki megaspore mother cell. Is the point clear? So nucellus e ovule form kore, tamane nucellus or cells bilak behave as megaspore mother cell. If I ask you what is the ploidy of the nucellus? Correct. Ploidy to ami janibo lagibo no. So ploidy of the nucellus ki hoy? Nucellus or cells bilake ki hisabe behave kore? Megaspore mother cell. Whenever we use this word megaspore or microspore mother cell, a mother cell or ploidy ki by any cell that undergoes meiosis tar ploidy ki twice n. So etu jodi twice n hoy, etu kune form korise? Nucellus e form korise. To nucellus or ploidy kiman? Twice n. Is the point clear? So questions ahibo ketiba nucellus or ploidy kiman. Later we will be studying structures like endosperm, endosperm or ploidy kiman, etc., etc. So while you are studying, ploidy is something that you should pay interest. Is the point clear? So ultimately megaspore mother cell form koribo, megaspore mother cell e meiosis hole keita form koribo, saitai form koribo, kintu saitar bitorot ki hobo, only one remains functional. I think I have said about this now before, je when we are using these two words, no, MMC বুলি কলে এটো কি হ'ব পাৰে? Microspore mother cell হ'ব পাৰে, right? If you are talking about the male part, or MMC বুলি কলে it could be megaspore mother cell also, yes or no? Mother cell, right? ই কি কৰিব? Meiosis undergo কৰিলে কি form কৰিব? Microspores, E ki form koribo? 4 haploid megaspores, understood? Out of this 4, all will survive. Out of this 4, only one will survive. I think it's clear. So this is all that we'll be discussing today. Next part of the lesson we'll discuss it in the next class. So next class or karne heading kori thoi dia types of ovules, right? Thank you. Ekin loke kiba doubts ase niki? Sai lua ebar.